A couple of weeks ago, I played a game of noughts and crosses, tic-tac-toe, and my opponent was DNA, and the DNA won. How is that possible? How can you play a game against DNA? There are these 9 wells. They represent the game grid, and in each of those wells is a special solution of DNA. And in addition to that, over to one side, there are all these other solutions of DNA that I can choose from. They represent the moves that I can make. The way we set it up is DNA takes the first move and plays the center square. When it's my move, suppose I want to play this square. Let's call that square number 4. So I find the solution that represents playing square number 4 on my first move, and I pipette that solution, just the right amount, into each one of those 9 wells. That's how I tell the DNA in those 9 wells which move I want to make. We then put those 9 wells into a special machine. Eventually, one of those wells starts to glow. Specifically, it starts to fluoresce, and that glowing well tells us which square the DNA has played. And then I choose my next move, I find the right solution, and tell each well by pipetting that solution which move I'm making. And then we put it back in the machine. The game progresses like that until there's either a win or a draw. Here's the cool part. The DNA will always play a perfect game of noughts and crosses. So the best I can hope for is a draw. How can DNA play a perfect game of noughts and crosses? Well, you can program a computer to play a perfect game of tic-tac-toe. So maybe there's a way we can encode that into the molecules of DNA. In a computer, it's just a series of logic statements like, if the human plays this square, then the computer plays this square. Every time the human makes a choice, the game branches, and you have to work out the logic for all of these branches. What's cool is you can take all that branching, flowing logic and flatten it down into a single layer of logic. You do that by looking at each individual square. So what is the logic of this square, for example? This square could become a cross for the computer under several different circumstances. Like after the human has played here, this would become a cross. Or later in the game, after the human has played here, this would become a cross. We don't have to worry about encoding when we are in the game. We just need one long logical statement full of ands and ors and nots related to what's currently on the board. This might not be the most efficient way to do it for a computer, flattening the logic down into each square. But in this way, we have a fighting chance of encoding that logic into bits of DNA in each one of those wells. If the human were allowed to go first, each logic statement would be horrendously complicated. By saying that the DNA goes first and plays in the second square, we simplify those logical statements and again, give ourselves a fighting chance. Let's have a look at the logic for just one square under these conditions. It's a whole bunch of statements strung together by ors like this. If this is true, or this is true, or this is true, or this is true, and so on, then the computer should play an X here. But what about these individual statements between the ors? Well, this means if the human played square 2 on move number 2 and square 6 on move number 3 and not square 1 on move number 1, then this square should be an X. You'll notice that we're limited to certain types of logic statement, like AND statements, if A and B. We also have at our disposal if A and B and not C. So how can you make logic gates out of DNA? Well, I don't. This is where my friend Andrew Holding comes in from York University. This whole thing was his idea, actually. He showed me this paper where these researchers had figured out the perfect structure for these little logic gates. We can just copy the molecules that they found to work well. This is where the paper talks about the DNA logic gates, and to explain it, I've turned these diagrams into physical models. So this is a little snippet of DNA. It's called a deoxyribozyme or DNAzyme because it's an enzyme built from DNA, which is to say it's a piece of DNA that can catalyze a chemical reaction. It can do something to something else without changing itself. And with a bit of extra mucking about, we can turn it into an AND gate. But before we do that, how does this catalyst actually work? You'll know already that the 4 bases of DNA, A, T, C, and G, pair up in a specific way. A always pairs with T, and C always pairs with G. And you'll notice that this sequence of A's, T's, C's, and G's pairs up perfectly with these A's, T's, C's, and G's here. So this strand of what they call the substrate attaches to this DNA catalyst, which then snaps it in half, and the bits float off on their own. So that's why this thing is called a catalyst. It's a molecule that can snap another molecule in two. You'll notice that there are these weird blobs on either end. This blob is called TAMRA, and it's fluorescent, which means if you shine light on it of a particular wavelength, then it will emit light at another different wavelength. This blob on the other end is called a quencher. It's like a little vampire that sucks energy from the fluorescent molecule. So when this molecule is nearby and you shine light on this molecule, hoping for it to fluoresce, instead, what happens is the energy gained by this fluorescent molecule is sucked into this molecule that then releases the energy as heat. So when this molecule is intact and these two blobs are near each other, you don't get fluorescence. But when you add this DNA enzyme into the mix and snap this thing in half, the fluorescent molecule can actually fluoresce, and that's something we can detect in the lab. To turn this into an AND gate, I'm going to add a loop like this and then extend it to cover this up. And I'm going to add a loop like this and then cover this up. And by doing that, I've disabled the catalyst, because look, when the substrate comes along, there's nothing for it to attach to. But what if we now create two more DNA molecules that pair up exactly with this loop and this loop? These will be like the inputs of the AND gate. Let's call these inputs molecules A and B. What happens when I add A? Well, it starts to attach itself to this loop. And as it does so, it's tugging on the loop. It's trying to straighten the loop out. These bonds here are now under strain, and eventually they will give way, exposing these bases. When you add input molecule B, it opens this side and exposes this bit of DNA molecule. So, with both A and B present, both parts of the catalyst are exposed. The substrate is snapped in half, and we can detect this has happened through fluorescence. And that's our AND gate. If molecule A and molecule B are present, then fluoresce. To make an and not gate, we add a third loop here. So let's introduce a third molecule C that pairs up perfectly with this loop. So when molecule C is present, it unzips this loop. These two bits of DNA end up far away from each other, and they can no longer act as a catalyst to snap the substrate in half. So this molecule is therefore an and not gate. If molecule A is present, and molecule B is present, and molecule C is not present, then fluoresce. These input molecules represent the moves that the human can make, and there are 32 possible moves, and these different wells need to be able to respond to those different inputs. So we need 32 different distinct strands of A's, T's, C's, and G's, and then pair those up with the loops around the logic gates to create all the logic that we need. You might be wondering where these ORs are coming from. Well, they're implicit ORs. Like when you have a bunch of different DNA logic gates in solution together, they don't have any effect on each other. Meaning if one of the gates activates and starts catalyzing the substrate, we will see fluorescence regardless of what the other gates are doing. That's where the ORs come from. Here's the thing though, to prepare these different solutions, and the 9 wells took hundreds of pipette operations, tiny amounts of liquid that we're transferring here and there. If we make any mistake, the computer isn't going to play the right move. And you can't test it until you've finished it all. So the challenge for us going into the game wasn't, can we beat DNA at tic-tac-toe? It was, have we done every single thing correctly? Before we find out, let me tell you about the sponsor really quickly, because this is an interesting one. There are these companies called data brokers that collect data about us and then sell that data to other companies. We don't know what companies they're selling our data to, but what we can do is contact them and say, stop, and delete my data. The problem is there are thousands of them, and they all want to be contacted in different ways, and sometimes they'll reject the request and you need to figure out how to respond to those rejections. Wouldn't it be great if a bunch of people did all that legwork, figured all that stuff out for every single one of them, and worked out how to respond to their rejection emails and all that sort of stuff, and automated it? Then the next person who wanted to do the same thing could do it just like that. And that's what the sponsor of this video does, Encoding. You sign up to Encoding, you give them the right to work on your behalf, and they just get to it. And you can see on the dashboard, look, these are all the companies that they've contacted, and this is the progress so far. And you start getting these emails from these companies saying, we've deleted your data. You know what it's like, you'll sign up for an email newsletter and you get a whole bunch of other emails from other companies, or you'll sign up for a loyalty card and then you start getting phone calls from other companies. I don't want that. And at the same time, I know myself well enough that I'm just never going to sort that out manually. Like, it would take months anyway because there are so many of them. From my point of view, it's an impossible task. And in that sense, I'm like the perfect demographic for Encoding. And maybe you are too. If you want to find out more, it's worth looking at the promo that we've got on this one. If you go to encoding.com/science, the first 100 people will get 20% off. The link is also in the description, so check out Encoding today. By the way, where do you get these custom bits of DNA? We certainly didn't make them ourselves. In fact, they're kind of off the shelf. Like, not in the sense that you could go to H&M and buy some, but there are companies and they have websites, and you log on and you say, can you send me this exact sequence of DNA, please? And can you send me this exact sequence? So we spent £2,000 buying many little sequences of DNA, and we had a go. So we played the game 4 times simultaneously on this grid of wells. We told the DNA our first moves by pipetting the right molecules into all the wells. Then we put the plates in the machine. 15 minutes later, we had a look at some line graphs that showed us how the fluorescence was changing in the different wells.
So now this one's coming up. So we've got to find out which well that is. It's gone in well 8.
Yes. Yes. That's what we wanted. That's amazing. Look. So that was this game here. It starts in the middle. I went there. It went to L8. And then we're going to go there. That's incredible.
Amazing. I mean, it was always going to work.
It was always going to work. Why did we doubt it? By the way, throughout this video, I've been referring to DNA playing crosses and humans playing noughts, but in the drawings you just saw, here they are, I've got the DNA playing noughts. Later on, I switched the drawings to DNA playing crosses. Sorry for the confusion. Anyway, here's game number 2.
So it's well 4 is a contender for it. It's gone, or well 6.
It was supposed to be well 4, and then I would play well 6.
Okay.
Well, that's interesting that well 6 came up.
That's almost certainly a pipetting error at some point.
Right. I doubt it would be me that made a pipetting error.
Your pipetting is flawless.
I love that this is the approach. Stick your finger on it.
Well 4.
Well 4. Well 4, it works. Look, there you go.
Well 8.
8. And it is 8. So that's amazing. So they all essentially work. There's a bit of ambiguity on the second game. So after finding out the DNA's second moves, we pipetted our second moves into all the wells and put them in the machine.
It is number 3rd highest, so I'll give it.
Yeah, great.
We just tried to...
This is amazing. So it took that move, as expected.
6 is lower than 4 and 9.
Yeah.
So whilst it's trying to cheat, it's hedging. It's hedging. It's like, didn't get away with it.
Didn't get away with cheating this time, DNA. All right, let's have a look at the last 2.
And we're expecting it to go in 1. Ooh. It's definitely coming up, though.
It's coming up, so that's good.
Ooh.
And it's, what is that one?
It's well 6, I think, shaky.
Yeah. We did a bad job on well 6. Final one. What we expect to be high is hopefully bottom left.
Well 7 hasn't really kicked off yet, has it?
Yeah.
It is going up.
So that'll carry on. It'll carry on playing that move, so to speak, during the next round. So for the next round, for this one, I'm going to play to win. So I'm going to go here, and then we expect the DNA to go here. On this one, I'm going to play there to block it, because that's now a naught. On this one, I blocked the straight down. So on this one, I'll block the diagonal.
So well 4 is not amazing.
Let's see it again. Sorry, I wasn't there.
There, so it's the top of the... Look, I guess that makes sense, actually. It's coming up.
It is. It's come above the pack, hasn't it?
So, yeah. And it is, I think if you look, see, it's below the dotted line here, and it's going above it here. It's slow.
So next, this is the one where it sort of took 2 turns, but we've kind of put it in its place.
So I think it's that one. Yeah.
Yeah. Yeah, come on.
Good.
Amazing. So this is the next one we're doing.
Well 9. Which is low.
That is low, but it's coming up though. Can we compare it to the pack?
So let's use my finger, high-tech finger.
Yeah.
Yeah.
That's definitely reached above the rest, hasn't it?
So we got
Wow.
Yeah.
So that's a lose for me.
You've lost.
Yay! Beaten by DNA. This one as well should also be a lose for me. 9 it should be. It's
And
coming
that's
up.
the problem we have, we don't give them
Yeah,
enough time,
we haven't given it enough time.
but if we look at that compared to the pack, it's now at the top.
It is, yeah, it's at the top of all the others. And if we gave it longer, it would come up further. But it's above everything else, so we know that that's a positive. So there you go. Isn't that amazing? Wow, so I've lost another game. So these ones should be a draw, which means we've still got to make another move on these. I'm going to move here to block this for my 4th move, and here I'm going to go here to block this on my 4th move, and those should be draws. So the way we're coding it, this is 6-4 because it's the 6th square on my 4th move, and this is 8-4 because it's the 8th square on my 4th move. So we're going to grab 6-4 and 8-4 from here and fill them in to the appropriate top 2 game boards in here.
Where do we need to go to?
It needs to go into bucket 9.
Yeah.
Yes. So it's above the pack. That's basically playing that move. And that's the end of the game. And this one, we expect it to play in position 7. This may not have worked.
So how many should be above? How many X's are there?
So there should be 1, 2, 3, 4 above it, above this one, because they've had longer. Then there's this rogue one as well.
Yeah, so 1, 2, 3, 4. That is the 6, and there's our 7. So this is the pack down here. So, I mean,
so
6
just about.
We definitely have a problem with our well 6.
Yeah. But it's not a terrible problem, actually.
And if we give it time, I hope this will move up, where this is actually kind of stuck where it has. So it's well 6 causes the problem. It's never really...
It's never taken off.
It was a sneaky cheat, by the way.
It was a sneaky attempt at cheating from the DNA, but it never took off. So there we go. We'll write that in. And that's the 2nd draw. So it worked. We created several solutions of DNA that encoded logic to play tic-tac-toe. And it played the way it was supposed to play. I was unable to win. Why do this at all? It's not about computing power. It's not about massive parallel computing on the molecular scale. It's about the interface between logic and biological systems. So in the future, maybe we could create sophisticated PCR tests that test for the presence of this and not this, for example. But just generally, figuring out how to manipulate DNA in this way is useful for all sorts of research relating to genetics. So far, we've only looked at a single layer of logic, which means we're limited in what we can implement. Researchers have implemented a full binary adder in a similar way before, where you have these molecular inputs and the outputs are fluorescence. But what makes a full binary adder useful is when you chain them together, so the outputs of one become the inputs of the other. But if your output is fluorescence and your input is a molecule, then there's a disconnect there. How about if, instead of the output being this molecule that fluoresces, it's a molecule that becomes the input of the next step in the logical sequence? Now that would be really interesting. A huge thank you to Andrew Holding and the University of York for all their help with this video.